Fermented Apilarnil with SCOBY, microbial biotransformation, cupric peptides with systemic and aesthetic effects. Apilarnil, a Romanian apicultural product invented by engineer Nicolae Iliescu. Obtained from drone larvae, 7 to 9 days of development, is recognized for its high density of proteins, essential amino acids, bioactive lipids, sterols, minerals, copper, zinc, magnesium, iron, phosphorus, chromium, and growth factors. In its raw form, the bioavailability of these components is partially limited, and their stability may be reduced. Controlled fermentation with SCOBY, symbiotic culture of bacteria and yeast, constitutes a biotransformation strategy that profoundly modifies the biochemical profile of apilarnil, leading to a bioactive complex with extended functional properties, biotransformation of proteins, and formation of cupric peptides. During fermentation, the enzymatic activity of yeasts and acetic bacteria determines the hydrolysis of complex proteins from apilarnil into small molecular weight peptides and free amino acids. These peptides are particularly rich in histidine, glycine, and lysine, amino acids known for their ability to chelate divalent metal ions, especially Cu2+, naturally present in apilarnil. The resulting peptide copper complexes are bioavailable and less toxic than the free copper ion, being preferentially used by cells, resembling the effects created by GHK-Cu. Benefits of cupric peptides: copper transport function, biological carrier. Peptides chelate Cu2+ and deliver it intracellularly. Increase bioavailability and reduce the toxicity of free copper. Act as a cofactor of cupro-dependent enzymes, supporting their catalytic function. Lysyl oxidase, stabilization of collagen and elastin. Cytochrome C oxidase, mitochondrial respiration. Superoxide dismutase, powerful antioxidant. Ceruloplasmin and peroxidase. 2. Oxidation of iron to Fe3+, the form that binds to transferrin for transport. Tyrosinase supports the transformation of tyrosine into melanin. Dopamine beta hydroxylase converts dopamine into noradrenaline at the level of the nervous system. Tissue remodeling signaling, damage response signaling. The peptide fragments resulting from fermentation are recognized by cells as regeneration signals. These activate fibroblasts, which secrete collagen, and modulate the activity of metalloproteinases, blocking their degradation. The result, active dermal remodeling, supporting the synthesis of collagen and elastin, without the formation of excessive fibrosis. Modulation of gene expression and cell proliferation. The peptide complex, together with the bioactive factors of the larva, ecdysteroids, sterols, can influence signaling pathways involved in regeneration, such as WNT/beta-catenin. The effect, anabolic and regenerative stimulation of fibroblasts and keratinocytes, with a reorientation of gene expression toward a more youthful profile. Control of inflammation and oxidative stress. Cupric peptides contribute to oxidative balance by supporting the activity of superoxide dismutase, SOD, and other endogenous antioxidant enzymes. They reduce pro-inflammatory cytokines, IL-1, TNF-alpha, and facilitate clean tissue healing without excessive fibrosis. Systemic benefits. Overall, the cupric peptides from fermented apilarnil support tissue regeneration and skin health, improve mitochondrial metabolism, contribute to oxidative homeostasis, act synergistically with the other bioactive compounds. Non-cupric peptides, organic acids, lipids, and postbiotics. Non-cupric bioactive peptides and immunometabolic effects. In addition to cupric peptides, fermentation generates a wide range of bioactive peptides without copper, which can be chelated with other minerals, Zn, Mg, Fe, chromium, etc., found in the larvae. These can exert immunomodulatory effects by regulating cytokine production, anti-inflammatory effects by inhibiting NF-kappa B pathways, antimicrobial effects by destabilizing bacterial membranes, neuromodulatory effects by influencing the gut-brain axis, complex adaptogenic effect through the bioactive composition, which is supported by lipids, sterols, and natural hormones from drone larvae. Organic acids and microbial metabolites. SCOBY produces organic acids such as gluconic, glucuronic, and acetic acid, well documented in acetic fermentations. These compounds contribute to hepatic detoxification, glucuronic acid, regulation of digestion and mineral absorption, gluconic acid, modulation of intestinal pH, inhibition of the development of pathogenic microorganisms. In addition, fermentation generates digestive enzymes, invertase, amylase, maltase, protease, lipase, catalase, phytase, vitamins from the B and K complex, and postbiotic metabolites, which support the function of the intestinal microbiota and systemic energy metabolism. Transformation of lipids in sterols. Apilarnil contains phospholipids, polyunsaturated fatty acids in sterols. Fermentation increases the proportion of bioactive free fatty acids, increasing their bioavailability and supporting cell membrane resistance and function, hormonal signaling, neuronal function, and neuroprotection. Additionally, the fermentation of apilarnil with SCOBY produces odd-chain fatty acids, C15:0 and C17:0, through microbial biotransformation. These acids have multiple beneficial effects on metabolism, cardiovascular health, mitochondrial function, and immunity, and can provide additional cellular protection against iron-induced oxidative stress, ferroptosis, by increasing cell membrane stability. A key aspect is the high intake of functional lipids from apilarnil, which provides the body with precursors for oxalipin synthesis. These lipid mediators have a wide range of biological effects. In addition to their well-known anti-inflammatory and pro-resolving roles, they contribute to cardiovascular and metabolic balance by regulating vascular tone, platelet aggregation, and blood pressure, supporting mitochondrial function, fatty acid oxidation, and insulin sensitivity. At the same time, they reduce oxidative stress, stabilize cell membranes, and can limit lipid peroxidation, including in the context of ferroptosis. They also modulate neuroinflammation and respiratory function, support renal perfusion and hydroelectrolytic balance, and participate in the microbiome immunity dialogue in the maintenance of intestinal barrier integrity. The SCOBY-type fermentation of apilarnil generates a complex postbiotic matrix capable of influencing lipid metabolism in the prostaglandin axis, PGE2/15, PGDH, through a series of synergistic mechanisms: modulation of the prostaglandin axis, PGE2/15, PGDH. The postbiotic complex contributes to lowering PGE2 levels and supporting a controlled inflammatory response in tissue regeneration processes, increasing the bioavailability of lipid precursors. Through the release and absorption of polyunsaturated fatty acids, PUFA, direct precursors of oxalipins. Anti-fibrotic effect through the action of isorhamnetin. The presence of bioactive flavonoids, especially isorhamnetin and luteolin, contributes to anti-fibrotic and anti-inflammatory effects. COX and LOX enzymatic modulation. The resulting postbiotics, organic acids, microbial metabolites, and polyphenolic derivatives contribute to the regulation of the COX and LOX pathways, guiding the conversion of PUFA toward a more balanced oxalipin profile with a reduction in pro-inflammatory mediators such as PGE2. Metabolic and endocrine synergy. Fermented apilarnil provides sterile precursors and bioactive compounds that can support endocrine homeostasis. Activation of endogenous cellular regeneration mechanisms. The postbiotic matrix provides a spectrum of active molecules involved in cellular signaling, supporting regenerative processes. Through functional reprogramming, it contributes to supporting hematopoiesis, tissue regeneration, metabolic balance, and neuroendocrine functions. Support of the intestinal microbiome and activation of functional axes. The intestinal microbiome represents an autonomous metabolic and immunological organ involved in the regulation of the majority of the organism's functional axes: gut-brain, gut-liver, gut-skin, gut immune system, etc. Fermentation of apilarnil with SCOBY generates postbiotics and bioactive peptides that can serve as a substrate for beneficial bacteria. Lactobacillus, bifidobacterium, Akkermansia, supporting the production of short-chain fatty acids, SCFA, butyrate, propionate, acetate, intestinal barrier integrity, and reduction of permeability, reduction of systemic inflammation by limiting the translocation of endotoxins, LPS. SCFA and bioactive peptides influence the functional axes of the organism. Gut-brain, regulation of neurotransmitters, neuronal plasticity and stress response. Gut-liver, hepatic detoxification and reduction of inflammation. Gut immune system, balance between the inflammatory response and immune tolerance. gut skin, reduction of cutaneous inflammation and support of dermal regeneration. Thus, cupric peptides and the other bioactive metabolites interact synergistically with the intestinal microbiome, integrating their effects into a coherent and adaptogenic systemic response. Integrated systemic effect. By combining cupric peptides and the other bioactive peptides, organic acids, modified lipids, and postbiotics, Apilarnil fermented with SCOBY acts as a multifactorial complex with effects on tissue regeneration, immune function, energy metabolism, oxidative and hormonal balance. This multifactorial profile differentiates it from classical supplements, offering an integrated bioregulation starting from the intestine and propagating the effects at a systemic level. Also, as a result of fermentation in SCOBY, the following are also formed: bacterial nanocellulose, which functions as a carrier for the active principles, exosomes, which support intercellular communication, tissue regeneration, and modulation of inflammation. Bacteriophages contribute to maintaining the balance of the intestinal microbiome. Acetic acid produced during fermentation, which by cleaving phytic bonds releases phytic acid, thus facilitating a more efficient absorption of essential minerals. Conclusion. Fermentation of Apilarnil with SCOBY transforms a traditional apicultural product into a natural bioactive complex with increased bioavailability and integrated systemic effects. Cupric peptides, bioactive peptides, organic acids, bioactive lipids, and microbial metabolites interact with the intestinal microbiome to support tissue regeneration, immune homeostasis, metabolic balance, and the adaptogenic function of the organism. This approach combines functional nutrition, fermentative biotechnology, and systemic health modulation.